So Glenn started as a PhD student at Columbia with Dave Reitman, where he was working on basically glass physics, like understanding slow dynamics, supercooled liquid. And as exciting as that field is, he decided to broaden his horizons. And so he went to do a postdoc in Chicago with Greg Voth and Aaron Dinner, and he did really cool stuff there, basically advancing multi-scale methods, coarse grain models, connecting the molecular detail with the cytoskeletal filaments to their collective emergent behaviors, something we all care deeply about and don't know well enough how to do. And then he started a few years ago as an assistant professor at NYU. He's continued to do really cool biophysics problems there. So interesting stuff on actin assembly, cell adhesion. He's also broadened out to work with the experimentalists there on colloidal physics, which is I think what we're going to hear about today. And so let me just say, I just love the stuff that Glenn does. He makes methodological advances in multi-scale models. He comes up with clever coarse grain models, and he puts those things together, and he learns, physics that are governing complicated self-assembly and self-organization problems, and makes advances in understanding how biophysics systems work. So it's awesome. I'm excited to hear what he's going to tell us about today.
Thank you, Mike. That might be overselling it a little bit, but we'll do my best. We do have a lot of overlapping interests, and as well as with people here. So I've been giving a lot of talks this year, as people often do in their 5th, 6th years as faculty members, and I usually have to talk about my proteins and molecular dynamic simulations and sampling configurations, which is what most of my group does. But a good chunk of people are working on self-assembly of colloidal particles, and I don't get to talk about it as much. So I'm excited to come here where everyone knows all of the things I'm going to tell you already, and then we can have a good discussion about it. I don't know if I can make this bigger because it didn't show my screen, but so if you can't see things, then let me know. Okay, try to click. Okay, so here's some movies of the 2 systems that I'll tell you about. One is a model for DNA-coded emulsion droplets, which I've been working on in collaboration with the Burgess lab at NYU. Another is about self-assembly of oppositely charged micron, or somewhat like micron-sized colloidal particles that we've also been modeling. But today I'll tell you mostly about the background to that and a very recent paper that came out last Monday about imaging those particles that some of you have already seen, but I think it'll be fun for discussion too. So before I get into it, I just want to thank my group. This is a picture from Tuesday, so it's like really, really fresh. That's a bonus for you because normally, we only get a new group picture every 6 months or something. That's pretty good, right?
For about 6 years.
Yeah. So yeah, it's fresh. So this is today. What I'll tell you about is a lot of people have contributed to, but Gaurav, who's in the back here, which I'll point out, worked with Yasna a lot for most of his PhD. He just graduated. He's going to WashU as a postdoc, and then Sanjeev is a postdoc who's been working with Stefano and some other people here, Michael and Nicole and Philip, who sadly got cut off by the waiter who took the picture, also contributed to some of this, which I only noticed later. And I'll also mention some work that I connects to these things with Liam Holt at our med school, and then these 3 are faculty at UT Austin that we have a new grants work on some assembly stuff too, which you'll see. Okay, so just to give you some other stuff, tell you about a little bit of other stuff that we're doing for a second. As Mike said, been doing a lot of work on how to quantify biomolecular ensembles and calculate transitions between relevant states of biomolecular assemblies, some of which is multi-scale in the sense that we can tackle this at the atomistic length scale and then try to use simpler models to understand more biophysical problems. To tackle these things at the molecular scale, we've been developing a lot of simulation methodology to bias to force these systems to undergo large conformational transitions, to calculate free energies and rates and things like that. Today I'll tell you about this aspect of my group involving self-assembly. And then we've also done some work on machine learning, like everyone else, especially on language models and what they can do for chemistry and chemical engineering that you might be interested in, but I don't have time to talk about that today. Okay, I have now getting to colloids. Please be kind, because I'll give you this introduction that you already know everything. But I hope you'll see like the thrust of what we're getting at, even though many of you work on this as well. So this, some of you have met Ted Huckle, who was a graduate student working with Stefano. And this is from this review article that he spearheaded. And a lot of the motivation, as you know, for studying things at this length scale of having large particles in solution that assemble into structures is maybe to understand more about the molecular world. So what can you build if you have the same kind of principles as how molecules assemble, but at the length scale where you can actually see things happening. So that's one of the motivations of using these colloids. And obviously you'll see in here that that is what's going on here. We'll be able to visualize everything in experiment that we're looking at. And so this is sort of a periodic table of different colloidal shapes that you can make. And one of the things that's highlighted, especially in this top row, is that you might want to be able to control. So first of all, you have to assemble them. Forgot about that part. Many of you here work with DNA, so you know that one of the ways you can assemble them is to coat the particles in single-stranded DNA with a sequence that you like, that has a complementary sequence or is self-complementary, then they'll assemble. And what I was going to say is that the thing that's highlighted here is a primary feature of how molecules assemble is that they can have valence, meaning that they have a preferred number of neighbors. And so if you want to build a targeted structure, you might want to be able to control that valence. So I've already talked to people here, which I also knew about the nice work, for example, being done with DNA origami and shapes. And that's another one context where people here are assembling things with specific valence and patterns of interactions. This is an example from my colleagues Marcus Beck and Dave Pine, where they put little patches on top of micron scale particles that they could then functionalize with DNA, try to assemble higher order structures. Okay. So this involves specifically designing certain building blocks, and I'll come back to other ways of assembling particles later. But one idea that's about 10 years old came from work that Jasna did, as well as people in the Netherlands, which was to think about if instead of having particles with DNA immobilized in a fixed way on their surface, what if they could move around? What would happen? And so these are works where in Jasna's group, what they did is they made monodispersed emulsions of oil droplets, which they could then functionalize with DNA on the surface. So it's a hydrophobic oil, and then they attached DNA to a hydrophobic moiety, which I'll also have a picture of later, and then it embeds in the oil and just floats around on the surface. And in this other group, they used a supported lipid bilayer on the surface. And either way, what you can get is tunable valence that depends on the stickiness of the interactions. So right after these papers were published, Don Frankel did some modeling work that showed that as you change the interactions, or as you'll see later, like equivalently the concentration of DNA on the surface, you can go from a gas to an assembly that really prefers to have a fixed number of neighbors, which has to do with sort of the chemical potential of having no bonds or many bonds. So this is really cool, and something that they wanted to understand more. And right when I started at NYU in 2018, Angus McMullen, many of you may have seen this paper. What he did is he systematically studied this oil droplet system as a function of concentration of DNA on the surface. This is the way that DNA is actually put into the oil droplets. So for those of you who are more experts, you can appreciate that. And what he found is that as a function of the concentration of DNA on the particles, you had tunable valence. And in particular, one of the situations they were interested in were these things they call colloidal, which are long polymers of droplets, which are they then showed were freely jointed. And these ones I always forget to mention. This time I think I remember. These are AB polymers, right? So you have complementary DNA, then you do a 50-50 mixture, and they self-assemble into chains. So if you have all self-complementary ones, they're much more likely to form a gel sample. Okay, but anyway, as you tune the concentration, you get different preferred number of valence from low concentration of DNA on the surface. Entropy favors just having a gas, and at high concentration, then you want to have lots of neighbors to satisfy as many DNA bonds as possible. You get a gel, and in between you can have these colloidal. So we wanted to understand, see if we could understand, what are the control parameters that give you this, for example, valence 2, or to tune this valence as a function of concentration and the other properties. I didn't realize at the time that, when somebody writes an interesting PRL, that like all the theorists in the world want to understand it. So there's many people who've been modeling this, including I saw one of the people here actually worked on this with Mognetti, but many people have worked on the theory of how these things should bind together and how they should fold and other things. But I'll just tell you what our attempt was. And maybe this is because I have like the same predilection as Mike, which he already mentioned, but I really wanted to build like a physical model for how this would happen, and then see what all the tuning parameters give you as you change them, in addition to, like, it'd be nice to have a theory, but I tried to, along with my student Gaurav, build a model of the system. So here's what we wanted as the minimal ingredients to the system. So we knew that we needed to have the oil droplet, obviously, and should exclude volume. And then we wanted to have something on the surface which represents DNA or binding sites. And we chose, but it's not really necessary to have 2 particles, because this is supposed to look a little bit like the real setup where you have a spacer molecule and double-stranded DNA, like PEG and double-stranded DNA, and then you have single-stranded DNA coming off, which is really like the handle where the bonds really happen. And the spacer part can give you some excluded volume that you can tune, which we're doing in other projects. But anyway, then if you attach the inner particle of those 2 to the center of the droplet with the spring, and you attach them together with the spring, and then you just do molecular dynamic simulations, they naturally undergo rotational diffusion on the surface. So that's cool. That's probably what's happening in reality. And then what happens when you put 2 of them together? Well, then you need to have some interaction between these outer particles. What we decided to do was to explicitly add and remove bonds through a Monte Carlo approach, where we can tune the rates of binding and unbinding. So sometimes a little hard to see, but there's a little extra bond in here that's explicitly added between these 2 particles. The reason that we wanted to add and remove explicit bonds is because in the situation, the real experiment with DNA, or what you'll see later, they can also put proteins on the surface, there really is a one-to-one interaction, and you don't expect attractive interactions with neighbors so much. And the other reason is we really wanted to be able to control these kinetics, although that won't really come up in the talk today. If you have a pair potential, which first of all, you have to deal with that it attracts many neighbors. So you have to make it like in the Frankel work, sometimes they make it angular dependent and very focused, or you can put like a little patch particle on the surface so that only 2 things bind. If people here have done that kind of thing, that's all fine. But we decided to try these explicit bonds so we could also control the kinetics. Okay, and so what's the approach that we have for that? This was a little inspired by the work that I had done on building cytoskeletal models, where we also had explicit bonds. So you want to run your molecular dynamics simulation, and then every n steps, jump into this other code that goes around and decides how many bonds should I add and how many bonds should I remove. And you want to have them added with some rate that's proportional to the on rate times the time step, like time interval that you're waiting, and then have them unbind with some frequency that's related to the off rate. And if you do this, you can, for a 2-state model of binding and unbinding, you can ascribe an energy parameter to this, which is basically the log of the on rate to the off rate. So it's the free energy of adding or removing an individual bond. And then this epsilon parameter is something we'll be able to control. And it's, you know, the same roughly thing that people here have been thinking about with the DNA, you know, 2 strands, and you ascribe some energy of interaction to those binding. What we did in addition to that is we want to have this satisfy detailed balance, meaning that the ratio of on to off rates for an individual bond is Boltzmann distributed. And you can do that by changing the on rates or the off rates or both. In this case, we decided to only change the on rates because we were thinking that we would make the off rates that depend on how stretched the bonds are later in a different, like, so that we could include slip and catch bonds, something I'll mention later. Anyway, so as a design choice, we just put it into this on rate, and then you get Gaussian here because we're adding harmonic springs. So basically, when you add a bond, it adds a harmonic amount of energy, and you have to take that into account. And then if you do this and you make all of your proposed binding interactions independent, which required a little bit of careful coding. So we propose a whole bunch of bonding events at once, but in a way that they're independent of each other. Then the whole thing satisfies detailed balance, and then you have an equilibrium binding setup. And so then you can have
this actually done on the GPU, or is that not what they're gonna get?
no. So yeah, that's a potential problem, but it turned out not to be the problem. Yeah, so what happens is if you change this n, which is how often you jump out of running code in HUMD, you can, you know, reduce the cost of that, right? So that it's no longer the rate limiting step. So we do like every 20 steps. The problem turned out to be when you run the simulation that I just showed, and I don't know if anyone wants to solve this, that would be great. For some reason, having tons and tons of harmonic bonds in HUMD is slow. We haven't really benchmarked if it's any slower than having tons and tons of non-bonded bonds, you know, like square wells. Should be fast.
Yeah.
Yes, I agree. So I don't know if it's slow, but that is what happens is as we do self-assembly, the simulations get slower and slower because we're adding so many interactions. And we're starting out with a lot of harmonic bonds because let's say that we have 100 particles on the surface, we have 100 bonds to the center and 100 bonds between the inner and outer particle and 100 angular
because you're
turns.
bringing all those things together, so now you have a lot of pairwise interaction.
I don't think so. Yeah, it seems to be just a lot of bonds too. Oh, well, it's probably both. Yeah. So that could probably be optimized, but we were able to get, you know, as far as we could get. And this is available, you know, on the GitHub page that goes with this project. But actually, for this project with people in Texas, we've updated it so it works with newer versions of that matters. Okay, so one other thing that everyone here is familiar with, and we did this right around when Ben visited, but later you'll see that in Yasna's group, they were doing temperature control over these DNA interactions. And so, you know, we wanted to include a way to reflect the fact that the binding interaction could be very steeply temperature dependent, as been measured by people here. So we can change the on and off rates. The way that we decided to implement that is with the switching function, where basically the on rate goes to 0 and the off rate goes to infinity as you go above the melting temperature, in a way that you can tune with a parameter so that you can have a kind of DNA-like melting curve. Yes.
Can I just take you back a minute regarding the speed problem?
Yes.
I was just wondering, do you do your simulations with single precision or double precision?
HUMD is single precision.
Okay, 2.9.7.
Yeah, but yeah, I don't think that's just as like that's not the major issue. Yeah,
because I've seen a sort of speed up with using, single. So when I was trying to do some new simulations, I was using double
And
precision.
that's specifically with the GPUs we're using right now, which have a lot of single precision cores and
Yeah.
double precision.
Yeah. But HUMD, this version of HUMD at least would have single precision.
Mhm.
It's just not as fast as I like, but if people are using lamps, it's way faster. It's like 100 or 1,000 frames, so there's at least that. Yeah, you'll see we get fairly big systems, but other questions? Yeah. Okay, so this will lead you up to a bit about so first thing we noticed when we were doing these simulations that was, I think, hadn't really been thought about in the experiments because they're not looking at the molecular level per se, is we noticed that when you see this recruitment of what's we call patch, it can happen in kind of two stages, which if you fit the recruitment to a double exponential, you get two time scales, one of which is the initial recruitment, but then there's also excluded volume, and that actually makes it take a long time for it to fully like course into a full patch. And so if you watch this, oh yeah, so the patch kind of looks like this, by the way, can look like this because of geometry, and people have worked with Yasna on like a theory for what the shape should be. We're still seeing if we can investigate that more. But you can see that at the end of this simulation, if you wait like 10 times longer, that this ring gets bigger and there's more stuff in it than here. And the reason that that is interesting or important is if you're going to have something which is kinetically controlled, which you'll see in a few minutes, we think that all these assemblies are, then it matters if particles are bumping into each other closer to this first time scale or second time scale.
So do they have experimental evidence for a ring?
Yes. Yeah, I'll have a picture of it later. I don't think it's right here. Yeah, you can they're fluorescently labeled and you can see that it's reaching. Yeah, but that depends on the surface tension of the droplet. So if the droplet is high surface tension, then it stays really spherical, and then it minimizes the energy for it to be in a ring and not like all of the surface. But if it's low surface tension, then it can deform and then you get more in like a pancake shape. Okay, so when we made this model, we had a problem, which was that we didn't know what to write about write a paper about. Because we could just write like, we made this model, but we really were searching around for like, what should we actually say about the system? And we decided to focus on what is a recipe, like how should you think about generating these chains, which may or may not be easy. And so that was our goal. We want to make this valence to most probable. And one of the reasons you might want to do that, which I'll come back to, is people are interested in these colloidomers because you can try to fold them up and make them as little patterned like tiles for bigger structures. Okay, but I'll get back to that. So how do you make long polymers? Well, we pretty quickly realized the issue with making long single polymers is avoiding branching, because then you don't have a single polymer anymore. So what are the recipe ingredients for that? First, you need, when two things combine, they can't all go into this patch. So if they all go into the patch, then you terminate your reaction at dimers. That would be bad. And when you add a third particle, what you need is that the center droplet has to get completely used up into these two patches. So you want essentially any amount could go in when only two things bind with each other, if it's reversible enough. But then when a third one comes, you really want it to split evenly between the outer droplets. And if you have that and then another one comes in, that property should propagate into having a polymer. So that's what we wanted. And so we tried to come up with is there a sweet spot for this? And there were three parameters that we could control at the sort of molecular level: the bond strength of the DNA, the radius of the droplet, and the number of DNA on the surface. So there's competing factors, which I always forget about explaining this in the right direction, but I'll just try to say the right thing. So basically there's like, how many can you crowd into the interface between the two droplets? That's controlled by like steric exclusion also, which is another parameter we could change. But also, so like the size of the particle, the bigger the particle, the more you can fit in because of the radius of curvature. But the bigger the particle, it favors things not going into the patch. So those are competing effects. And then when you increase the bond strength, more things go into the patch. So basically, you can tune all of these things and look at a dimer where you want to have, like I said, you want to have 50 to less than 100% and more than 50% go into the interface. And then when you have a trimer and you look at terminal droplets, you want it to be around 50%. and the inner droplet to have 0. So somewhere in here should be good. And you can change the number of binders under these conditions. And basically you want to pick the Venn diagram intersection of these conditions and then see if you get chains. So then there's another, oh, I'm sure this paper is published now. Anyway, there's another aspect that you have to think about, which is now when you start assembling, it's not just about the energetics, but you also full control. So you can change the volume fraction of all of your droplets, and you can also, we realize you can change the viscosity of your system essentially, which changes how often they bump into each other. Because as I mentioned, you might think this could be kinetically controlled, or that's what we thought. But interestingly, this volume fraction one is confusing because it both changes how often things bump into each other, but it's also a thermodynamic control. So that low volume fraction you expect to have basically a gas, and a high volume fraction you want to have a gel, and somewhere in between it might have these colloidal, but it also changes the kinetics. So we varied all these things, and then it turned out to be a really hard optimization problem. I was talking to some people earlier about like design problems. So here, this we thought would be a really good system for testing algorithms for, design, because we have like 5 or 6 things we can control, and how do you optimize things? And some of you are probably familiar with this problem where you have to optimize multiple things at once as well, being a problem. So we weren't really sure what counted as being the best colloidamer system anyway. Do you want the longest chains or the most chains or the least defects or other things? So we measured each of those, which I was thinking about, like when you choose your car in Mario Kart, you want to be like really fast or really, robust. So somewhere in the middle, probably. So you like try to find the one that's like the most like Luigi or whatever, and then that's like this one. So that's what we went with. We'll still play Mario Kart, right?
Got a good deck player.
Yeah. So okay, so if you do that, this one is now colored. This is a 50-50 mixture of A and B particles.
I got confused. What is Luigi? What do you actually
Oh,
optimize?
oh, sorry. Yeah, like you want to be like good at you want to be like pretty good at everything. So the things that we optimize, which I'll also show on the next slide, are so first of all, here things are now colored by motif. So we want to have you optimize the fraction with valence 2, meaning 2 neighbors. So that means we're taking away monomers as well. But so like you can say no defects, and if you really push for no defects, you can have a lot of dimers. Dimers don't have any defects, right? Because there's no branching. So we can have what's the which has the longest chain, essentially. So which condition produces the longest or the most extreme length distribution? It's not quite that. Which has the least mistakes and which has the most valence too?
You're trying to maximize all these things at once.
Well, we're not really trying to optimize them, or we just at once, but like, so this condition, which is a little higher viscosity, so I didn't say exactly what, so what you want, as people here probably have thought about, is, so you have 2 things come together, that's fine, then a third one comes in, that's also fine, you have A, B, A. Now a B one comes in, and now your big challenge is it gonna form a loop? Because if the ends come together, you have a square, and then you're done, right? Then you don't have a chain anymore. So if a 5th particle comes in, you have ABABA, then you're pretty good, because that I don't remember the exact scaling law, but many people here probably know more polymer physics than me. And the end-to-end collision distance scales as n to the something, 2/3 or 80%, or I don't know, something like that. And so the longer it is, the less likely your ends are to run into each other, right? So that is advantageous for us because we don't want the ends to link up in this case. So you can try to make it so that the time for that to happen is a little bit longer for the 4 than it is for a single particle, which moves faster to bump into your chain, which depends on the concentration of the viscosity. So
when you say viscosity, is the viscosity of the solution or the membrane?
Right, so we're actually controlling those independently, or I don't know what else we could control, but we have 2 drag coefficients in our simulations, which I didn't really say. We have one that's for the droplet, which is basically the solution viscosity. If you want, you could add methyl cellulose or whatever, and it would do this. That's the idea. But we don't, the DNA moves on the surface much faster, which we control separately because we assume that's what would happen in reality, but at a molecular timescale. So we control this. When we up this, it doesn't change the time for the patch to assemble. Yeah, so I don't know if that answered your question, Mike. We're trying to get all these things. So we didn't do an optimization algorithm. Gaurav ran 1,000 simulations and looked for the one that on average had the best of all three of these things. Yeah.
What's the difference between the top row and the bottom row?
This is the higher viscosity and this is the lower viscosity. So at the higher viscosity, basically you should have, at the same fixed amount of time, you should have less stuff happen up to that point.
Do you have any parameters that control an effective bending energy?
No, so these, like, I should say no, but essentially these are freely jointed chains. We can make it so that this droplet DNA droplet interaction has a torsional term. So and the way that we can do that, which is more to get back to this ring thing that Mike asked about, is right now we have the DNA when they're like in these simulations, I mean, when two things come together like this, it forms a bond so that you have like 4 particles and 3 bonds. Another thing that we can do is make it so that the rest length of that interaction is 0, and then there becomes an effective torsional angle across each of the DNA. So that would make them straight, but that doesn't necessarily make the backbone rigid.
Okay,
Exactly.
like droplet size has no effect on it.
No, it's supposed to end up being freely jointed. There's like some drag, I guess, which makes it on a finite time scale, maybe a little bit more rigid. Other questions? Okay, so what happened? This is colored by these motifs. So like whatever this color is, it's like the chains, and that's what we want. And these are defects. So you don't want that. So you can run for a while. You see that you get dimers and chains and only 1 defect so far. And then you can quantify those things. It basically happens in a stepwise fashion. One of the things that was interesting is that our condition had fewer defects than in the experimental version. Although in the experiment, I wouldn't say that Angus was necessarily trying to do his absolute best to get no defects or whatever, but we ended up checking that, which is interesting. But here's the problem. We found, so there's 5% of particles in here have valence 3 and none have valence 4. But if you have 5% of particles with valence 3, it makes your number of pure chains go all the way down to 50%, because every single particle with valence 3 like kills 1 chain. So if you have a chain of length 20 and then it has like 2 things sticking off it, doesn't count as a chain anymore, according to us. So that's a problem. And another problem, which is probably obvious to people here, is that when you have this kind of assembly process, then the length distribution is exponential. And so you can make, we got really long chains for our number of particles, like longer than the ones they had in these experimental conditions too. But you're not going to have like, there's no recipe to doing this to say like, oh, I want a monodisperse system of length 12 so I can study their assembly. So actually self-assembly is not like a great idea here to do that. And then here's the other thing, which is that after we had this slide, essentially, I said to Gaurav, like, you have to continue this movie and see what happens anyway. So if you keep watching this movie, you'll see that, in fact, or if you had really zoomed in and looked closely, despite our design parameters, it's not that 100% of DNA was in the patches, it was like 95%. And that means that there's still this possibility of forming occasional defects. And so you can really see from this movie that this was a kinetically controlled state anyway. One of our reviewers told us that there's no thermodynamic recipe for chains, which I'm not sure is true, but anyway, you can take that with a grain of salt anyway, but you can definitely see that, and I think Angus would agree that he did the experiments for 3 days or whatever, and then the results are whatever they were in 3 days. And that's the same with the simulation. So I think that's an interesting thing to keep in mind. But that's why that these binding kinetics really matter. Okay, So what can we do with these systems? Well, after we did this, Yasna had this other paper with Angus where they did folding of ABAB chains and homopolymers, and they looked at all the pathways, and that was really nice. We had also in this paper been interested in, could we study folding? The good thing is in simulations and also the experiment to a certain extent is you don't have to do self-assembly to make these chains, you just make them. So we can start with a certain pattern of droplets. To do folding, what we do is we now coat the particles in two kinds of DNA. One that's supposed to form the backbone at a, it has a melting temperature that's higher. Yep, so then when you cool below that melting temperature, it forms the backbone, and then you have another one that's lower, weaker, and you cool below that one, and it makes like the side chain interactions. So this is what that looks like. If you cycle the temperature, it goes to a disordered state, and then it collapses again as the temperature goes back down. So you can do a repeating cycle of that. And what we did as a design thing is there's 4 rigid structures, which I'll show, they're up here, but I'll also show them again in a minute. These ones that they call ladders and rockets and flowers. And Gaurav tried to see if he could find a condition where you get all of them in single bond simulation. So what does that require? Well, one of the things that's interesting is that this flower, so in that paper with Yasna, they looked at these kinetic maps and the pathways to get between things. Turns out that this flower, which is the lowest energy because it has the most bonds, most patches, in order to form it kinetically, you have to form one of these other structures, have a whole patch break, and then have it like swing around and form this flower. So you can actually see that in this energy trace here, where the energy is the same for all of these structures. And then in this section, it drops down as one breaks, and then it goes up to a higher, well, it's sort of energy. It's the number of bonds, I should say, on average. So it's like flat, 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 and then it jumps up to this higher one, which is cool. And getting that required a couple of design parameters involving having the particles be kind of small enough to crowd stuff out of the patches, and having the energy of the interactions be like 3 kT, stuff like that. But when we did find a recipe where we could get all of them at once, and then we looked at the yields of these rigid structures, they turned out to be quite similar to what was seen experimentally, although I don't think this is actually in their paper, but it's something they kind of quantified on the side for this system. So that was cool. And we learned a little bit about the rules for doing that. Okay, so some of the future work with this dynamic bonding model, and I can hustle through the other colloidal thing, is to look at these rings and what makes them. So here's the experiment, Mike, that's, doesn't look quite this clear, but, you can fluorescently label the things and quantify. This is how good the histograms are, I guess, of height distribution. And Sasha Hagenfeld at Illinois has a whole theory for how big these rings should be and stuff. So we're trying to understand if we can reproduce that in our model or not. But for some reason, it's not quite as clear as it probably should be. I don't know. We're still ironing out the details of that. But this is what it looked like in an earlier version of the simulation. So one thing we can study here that you can't get from a 10-parameter mean field model is the kind of behavior of this coarsening that depends on the parameters like the size of the particle and the viscosity and other things. So I think that's kind of cool.
How do those compare to the full and the assembly?
Oh, time scales. Yeah, I think that the full coarsening is longer. It's longer than the collision time.
No, but compared to the particles moving around. Oh, so
I'm saying like the full coarsening to like the maximum droplet size, especially when what this is like a 3-micron particle with 10,000 DNA. It takes the entropy really favors it not all going into the droplet, and like so coarsening takes a while, and so
longer than it takes those droplets to move around and find each other and make change and stuff.
I'd say maybe comparable time, because also when you get up to these big particles, then it takes a long time for them to run into each other too. Yeah, so I think that's something to consider in the design. So a lot of the work on the bio side has been looking at the force dependence of bonds, and one of the places that that's important is in adhesion proteins on the surfaces of cells. where you can have this surprising catch bonding behavior where when they get stretched, they actually last longer. And you can see how in that movie I just showed that could contribute to like the size of this ring and its assembly kinetics. If when it's at this edge of the ring, it's stretched out and it wants to be stretched out, that would like spread out the ring. So we're interested in how that affects this assembly. That's also relevant for understanding how cells interact with each other because they basically work in a similar mechanism where you assemble an adhesion patch of monomers on the surface that are mobile. So that's one thing that Yasna also works on in her group. And this is an example of that, where they put coherent molecules on a bunch of formable oil droplets and they squeeze them together to make them kind of like a tissue, and they let go. And they see how the force sensing of the coherence helps stabilize that structure. So that's something we want to do more. There's other things that we can do with this dynamic bonding model, and people might be interested in this too. So a different kind of self-assembly problem is that's very hot also at the moment is liquid-liquid phase separation in cells. And one of the theories for how that works is that you have particles that are multivalent. So it's quite similar to what I've been talking about. So you have in those cases, you have often people think of it as a sticker and spacer model. So you have a polymer with a lot of sticky spots, and then that will tend to form a liquid droplet. With Liam Holt at the med school and his postdoc Tong, what they wanted to do is separate out those physical principles from the biology, even though they're biologists. And so what they did is they designed the synthetic system where you have a hexamer with binding sites on the surface and a dimer with complementary binding sites on its surface, and then a little drug molecule that induces the binding. And so these should assemble. And from our physics perspective, right, you can make this sort of spherical Cal model of that, which they're totally into anyway. And so we wanted to have a model of this kind of assembly, which this is it happening inside of yeast cells. So we can assemble with this on and off binding kinetics, these patchy particles that have fixed valence. And the key thing that I didn't tell you that Liam's really interested in is how the crowded, dense environment of cells affects processes. So inside of the cell, there's like 30 to 50% of the volume is taken up by macromolecules. And so what we were studying here and what they studied in the experiment, in this paper, which is about to come out, is how the amount of crowding in the cell affects the nucleation and growth of these droplets. So the upshot in one slide is that if you can have realistic binding affinities like this, like so the proteins can have binding affinities of say 10 micromolar for each other, and then you'll have no assembly at 0 crowding. You add crowding to the realistic levels, like 30%, and it increases the nucleation rate by decreasing the amount of free volume available to little microscopic particles, and then you can get large liquid-like assemblies. But if you go to too high a crowding, which you can do in the cells by various mutations and osmotic conditions that they've characterized before, then what you get is you inhibit coarsening, so you get lots of little droplets. So that's the upshot of that paper. And then separately, the other thing we've been doing that I don't have a lot of new results. I just wanted to mention in this with these people in Texas, we've been looking at assembly of nanoparticles now instead of micron-sized particles. They're actually pretty similar in size to the proteins. And so we have this grant to study assembly of nanoparticles with fixed valence via bridging ligands that have reversible covalent interactions. And the reason that I got involved in that is that our dynamic binding code is pretty convenient for being able to understand the gelation properties of this. Well, you can do that anyway, but maybe you can ask about how does changing the kinetics of binding and unbinding change the, the rheological properties of the gels, for example. Okay, so stay tuned for that, I guess. Cool. So I guess I have 15 minutes or something, right?
Yeah.
Other questions?
Some time for questions.
Okay, so just to wrap up, we have this code that I think is useful of this model that we can study a lot of different kinds of self-assembly problems. And we learned a little bit about how to control kinetically the assembly, but we're still trying to learn a lot about the thermodynamics of formation of these patches and how we can do other self-assembly. And then, as you know, I think people here might be have some ideas on strategies for optimizing the conditions to get a particular outcome, which we didn't really or didn't grow up, didn't have time to pursue, but it's still like I think a good system for that. Okay, so getting back to this self-assembly problem, I'm going to tell you about the work I've been doing with the Sakana lab, and it started with Ted, who, as I said, some of you met, just collaborated with Ben. So as Stefano has told me, cheekily, I guess, like you can do a lot of stuff with DNA-coded colloids. So when he started his group, he wanted to know what can you do not with DNA-coded colloids. Like, can you do things with shape and other ways of changing colloidal shapes, like these lower areas that give you some of the same properties? And I think people here will really appreciate this. Like, one of the main reasons for that is just because it's expensive and lower throughput to use DNA. And so if you can avoid that, then maybe you can make bigger crystals that are like macroscopic. So it's well known, Ben's done this, other people have done this, that you can form binary crystals from, I don't know where that mouse came from. You can form binary crystals from DNA-coded colloids, and you can get different crystals by changing the size ratio and the number ratio of the particles. Lots of papers about that. And so many people here are more experts on that than me. This is an example of what I was talking about, although this is an older paper, like, but maybe you get crystals on this size scale with thousands of colloids. Okay, so Ted's goal was to make bigger crystals, and the strategy he came up with was to try to make ionic crystals, you know, like table salt. But what's the problem with that? And you know, this had really only been done once before by the Van Vleuteren group, because the issue is when you mix these two particles, or when you have particles in solution, I think a lot of people here already know this, there's something called the LBO theory, which tells you that the thing that prevents the particles from irreversibly aggregating due to van der Waals forces is their surface charge. So if you have repulsion between the particles due to their charge, then they don't get close enough to stick to each other. If you take away that surface charge, or you make them oppositely charged, then they stick together anyway. So how are you going to prevent that? So to make this crystal. So here's a movie of that's not that relevant, that Ted made, where this is supposed to show that you mix them together and it aggregates. Okay, so then you can do an SEM and you see you get like junk. All right, so this was Ted's idea. Some people here have been doing this, so you know it well. The idea is that the interaction between oppositely charged colloids in solution is modeled by basically a screen Coulomb interaction, where the interaction range decays very quickly due to salts in solution, and that's characterized by the Debye length. So basically you have surface charge, and then they attract or repel each other over a length which is in the nanometer scale. And then these particles are in the hundreds of nanometer or micron scale. So it's actually a very short range electrostatic interaction. And the key that Ted realized is that if you could, which is sort of already done, you can coat these plastic beads with a polymer, then that polymer might keep them, well, it could keep them separate, but the key is that if they're just the right length, then they'll be in tune with this Debye length, so that they just feel each other. And as you change the Debye length, then here, it will tune you from purely repulsive to attractive to irreversibly aggregating, and which you can see here. So this is aggregate, stable, and you can see little colors here reflecting the fact that it assembles into a crystal. Okay, so this was in this previous paper, and we did simulations. I'm mostly going to tell you about the experiments. Yeah, so this actually, that's kind of one of the nice things. There's lots of approximations in this theory, but you can see the upshot is a potential that kind of looks like a Morse potential. We can also simulate a Morse potential if we want. The key is that we kind of know to pretty good approximation based on our success of the simulation model that this is a good reflection of what's going on, and that means that we can also test things in simulation that are hard to test in experiment. Okay, so this is kind of an overview of showing the same kinds of things, that you can change the size ratio and get different crystal structures. But here, I don't know what the scale bar is, but these are faceted large crystals with billions of colloids in them, which is cool. So you can get macroscopic crystals, which you can see here, even with naked eye embedded in like a little gel. So that's good. Of course, you can't programmably assemble things like you can with DNA. So it's not like one-to-one, just to make that clear. But it's good for some things, perhaps. Good for making cubes. Okay, so this is what, as an example, what classical nucleation of these crystals might look like. And you can see that they're really nice under optical microscope, and they get big over a couple of hours. And we can also simulate that. You'll see that you get kind of similar behavior. And then I didn't have time to talk about this today because I knew I'd run out of time. But most of what we're going to do in our next paper is about non-classical nucleation and other pathways for assembly. You can get both depending on the condition. We're trying to understand that. But just to give you an idea of what it looks like. Okay, so we had this paper that came out last week. I just want to tell you about it because it's cool. Some of you have already seen it. Here was the deal was that you can do these simulations, and simulations, you can watch every particle and see what happens, but nobody believes the simulations. So you want to be able to see every particle and see inside the crystal experimentally too, so that we can prove what was happening in the simulations was real. And so we took this whole 3-year detour led by Shihao, who is a graduate student in Stefano's group, to be able to see inside these crystals. So also part of that is for identifying what crystal you get. I was asking Ben when he visited like a year or two ago, like, how do you know what crystal you got? And then it's like partly by inspection, right? But like by guessing and checking, which is still true. But anyway, just telling you that if you knew the positions of all the particles, which you do in some cases in your labs here too, then it's less guessing and checking. So that was the idea. So what Shihao was getting annoyed with is he would make these crystals. Yeah, a lot of times it's the cesium chloride like one, and he sees it, he knows exactly what it is just by seeing the facets. Oh, I should mention one other innovation, if you want, from Shihao, is that he really shrunk down the particles since Ted's paper. So he's been really pushing it to the limit of what we can see. So now working with like 200 nanometer particles, the assembly happens 10 to 100 times faster, and you get much bigger, or at least many more particles in a crystal. So we're starting to push up into the like almost centimeter scale, which Stefano threatened Shihao that he couldn't graduate till he had a centimeter crystal. I don't know if that's going to happen, but we're like, depends how you round. So anyway, he made smaller particles, they go fast, and then it's like, oh, well, what crystal did you get? Well, then you look at the surface of this crystal, which has extra gunk on the surface and whatever, and you look at these planes and you try to say like, oh, if I had this unit cell, what planes would I get on the surface? Which people sort of know, and then you can say, this must have been cesium chloride. But if you could look inside, then you could, check the unit cell. That was the goal. And so what he did, along with Adam Hauser, who was a postdoc with Stefano and Paul Chaikin, was to synthesize, instead of polystyrene beads, a fluorinated polymer, which is known to have to be much closer to being index matched with water. So I guess I didn't already say what the problem is. So you can fluorescently label these polystyrene beads, and then you can do 3D imaging. But because of scattering, you can only look a few microns in like 2 or 3 layers, 5 layers, which I probably have a picture of. So what you want is your particles to be index matched, in which case light won't scatter off of them. And that means you can do confocal imaging like all the way through the crystal up to the level of bleaching of your dye. So this is it. They made these particles monodisperse of negative and positive charge. That was hard. One of them had stable, I think, negative charge, and the positive ones were hard to get, but they figured that out. Adam's a really good polymer chemist. And then you can change in water, they're pretty index matched, you can also do a 40% DMSO and they're perfectly index matched. So here's what a crystal looks like when it's index matched, which is like ice, right? It's pretty cool. Okay, so here's like the kind of synthetic scheme to make positive and negative particles from this fluorinated monomer. And yeah, here's what you can see. So this is polystyrene, makes nice crystals. Fluorinated one makes nice crystals. And then fluorine, and they're kind of transparent. And then if you put it into DMSO, they're like invisible, but you know, they're still there. And we don't know why they're like a little bit more rounded edges. No idea. If anyone has any ideas about that, that would be great to know. But still really nice macroscopic crystals. And this is a line scan in the confocal imaging. And what you can see is that the height of this peak of where you have a particle goes down exponentially. And in polystyrene, it's gone, the signal, in 5 microns, which is for these particles like 10 layers. And in water, it's like 70. And in DMSO, it's like 90 microns, which is, so this is comparing 5 nanometers. I mean, 5 microns to 5 microns, like the clarity that you have in water in both cases. So, so that's really cool. And just for, since everyone here is like experts, or might want to use this, we are pretty sure that this depth is not just limited, it's not really limited by scattering, but it's mostly limited by bleaching. So the thing is that if you want to scan to the depth of 70 nanometers, then you've scanned at like 1 microns, 1 micron, 2 micron, 3 microns. So in all of those times, you have light going deeper into the crystal and contributing to bleaching, so that by the time you get to 90, you're bleached. But probably you could go deeper if you only cared about the other side of the crystal.
Probably.
Oh, yeah. So this should be, yeah, 2 more minutes. Okay, good. So here's just a simulation showing that things, if you have a surface, that things can crystallize on the surface. It looks like that. The reason that that's convenient is because a negatively charged glass surface, then we can do this 3D cross section of the crystal, and it just so happens, not just so happens, but because of that, the planes are oriented parallel to the surface, which is really convenient for imaging, right? And then we can, like I said, you can get the 3D coordinates, and you can do like scattering, and you can look at the different planes. So I'm just gonna, and then, you know, this is what he told me that you guys were doing as well. So then we tried to do that. Okay, and then just this is the last thing that in order to impress the reviewers, we also tried to look at something that was time dependent. So we looked to see at the defects inside of the crystal. Stefano was mad because he said his crystals were defect free. So, you know, it's not really pretend that there's no defects, but otherwise, if there were some, they would look like this. And then one thing that really surprised us, so what we did is an experiment where the salt concentration changes and the crystal melts. It melts by like a sublimation mechanism, and the vacancies never move. But if you have like a multi-level defect, then you can actually see the defects moving. So that's cool. And also in simulation. Okay, so that was it. We have this new system. It also matches the simulations. And now going forward, we're looking at using that technology to look at the nucleation and growth mechanism of colloidal crystals. Okay.
Why do you get a bigger crystal than particles?
Well, it's more, there could be other reasons, but I think it's just like diffusion limited and time. So you have just more opportunities for things to come on and off and rearrange when you have smaller particles. Sort of thing. I think that that's the main thing. Oh, and I can give you another reason, which is that the bigger the particle is, the faster it sediments. And so you get like much more pancake particles than you do with small particles, which have a higher gravitational length.
But you fix the volume fraction.
Try to, you know, every time I ask, like, what's your concentration? They're like, yes, but yeah, it should be around the same concentration, volume fraction. But that, yeah, it's I think partly the gravity thing and partly the collision thing.
When you're comparing or you're trying to characterize the structure of the interior of the crystal, do you ever notice like the surface of the crystal has like a different like it would give you a different kind of like structure than the interior?
Well, let me think about the way to answer that. So the surface, if it's a faceted crystal, which only happens for reasons that I'm no expert, I guess it's well understood. But when you get up to like a certain size, then it wants to form these facets energetically, and the facets should be cut out of a plane of the crystal. So in that sense, they're different structure than the bulk, but they're still formed from the bulk. A more sophisticated answer would be that in our new paper, we have data showing that like one plane of a surface can nucleate a different crystal structure. That's probably known to, like, which we are calling, or people call, epitaxial growth. So you could imagine templating a specific structure you want by making different structure. Like if a certain has certain, the surface has certain characteristics, it could create a new crystal that has different characteristics. That's what we
Thank you.
I guess, but as a maybe related question, you never see like surface relaxation, like there's no difference in the actual atomic structure of the atomic structure of the facets.
So I think that in the smaller crystals, that I guess maybe this also answers your question. So like another thing which definitely happens in nanoparticle crystals, I mean nanoparticles itself, like cadmium selenide, is that the surface can have like an excess of X ions. And I guess I should say that yes, I think that happens. So in this thorium phosphide crystal, which was in here, but you probably didn't see that paper, I think it's in there somewhere. That one, like when I asked Sanjeev, what's the ratio of positive and negative particle crystal, and you think it's 3 to 4, but it's not. It's like 80%, which suggests, well, or you could just count, but indeed, like the surface can be kind of terminated by the to match. Yeah, anyway, they can be terminated by an excess of one of the charges, but not really terminated. Keep growing.